back there. Wait, I also have to take this off because although it keeps me warm, it totally moves my outfit. Thank you to the survivors who are still here at this stage of the game. We are the survivors. As I've already sort of said to a number of you, this is not the talk that I kind of hope to give, but I hope that you'll find it interesting anyway. This is the title that I submitted, but this is actually a little bit more accurate about what I'm actually going to talk about, and I'll explain why. And the personnel on this are two graduate students, Dinusha and Alex, and then we had a REU student, Ahsley, and an undergraduate, Kevan Elkins, who is now actually working with us as a technician. And I'm going to give a little bit of background about the sort of broader context for the project, and then I'll talk about the transcriptome data a little bit. I am interested in this evolutionary phenomenon of the origin of fleshy fruit, which has happened many, many, many times during the course of angiosperm evolution, and is of course important economically, but also ecologically. And we're working in the Solanaceae because it is the best family. It is the best family. These are its attributes. Genomes have been sequenced. You can knock out gene functions, well-supported phylogeny. And it does have this key feature of having a transition from dry to fleshy fruit. So this is the basic phylogeny here. And at this point where the star is, there's a more or less of a wholesale transition from dry fruits to fleshy fruits. And so we can compare what's going on in this part of the tree with what's going on in this part of the tree developmentally to try to identify key differences. And what also makes it even better, I don't know if you can actually see this, is this branch just turned red, as did this one. We have two independent origins of fleshy fruit in Cestrum and Brunfelsia, and also a reversion to a dry fruit here in Datura. So we can not only look at what happened at the main sort of switch from dry to fleshy, we can also ask if, for instance, Cestrum sort of uses the same mechanisms to form a fleshy fruit as, say, Nicotiana or Petunia, and we can also ask whether Datura has sort of reverted to the same mechanism to form a dry fruit as Nicotiana and Petunia. Excuse me, the Cestrum and Brunfelsia as tomato, a fleshy fruit. Sorry about that, misspoke. Just to show you what some of these look like, on the top are three genera from the sort of dry fruit part of the tree, and the bottom are three genera from the fleshy fruit. And these are your typical capsules from Petunia and Nicotiana. And then here's the berry of Cestrum, which in many ways, in terms of sort of morphological or ontogenetic development, is more similar to the capsules of Nicotiana and Petunia. And then on the bottom, you have Solanum and Capsicum, which have the typical berry, and then Datura, which again has reverted to this dry dehiscent fruit, which again in the early stages of development actually is a bit more like a tomato and then kind of goes off the rails. So this is the system that we're working in, and we're focusing on these two species, Nicotiana obtusifolia, or desert tobacco, which happens also, in fact, to be kind of native close to where I now live. And if you're ever interested in working with a Nicotiana species, I highly recommend this. Its genome has been sequenced now, and it's much more compact than most Nicotiana species. It flowers profusely, and it flowers continuously. So we find it much more convenient than tobacco. Oh, and it's diploid. So tobacco, of course, is tetraploid, and some of the other species that are frequently used, like sylvestris, take forever to flower and get absolutely humongous. So that's my little sales pitch there. And then we're also working with tomato, and we're basically trying to identify genes that determine which type of fruit will be formed, and then what the functions of those genes are specifically in fruit development. And the project in the moment is focusing on the orthologs of the gene fruitful, and I'll explain why. So this is fruitful in Arabidopsis. I'm sure you all recognize the fruit of Arabidopsis here. This is a fruitful, the fruit of a fruitful mutant. And here you can see the scanning electron microscope picture of a wild-type fruit. And in this cross-section here, more or less, say, through this area, you see these patterns of lignification here. And in the fruitful mutant, this lignin is missing, and as a result, the fruit doesn't hiss. But the seeds develop pretty normally, and so the fruit actually ruptures prematurely. And so we have an idea of what the role of this fruit might be in a dry dehiscent fruit. We know what it is in Arabidopsis. We have a hypothesis for what it might be in Nicotiana, but we don't actually know if they're serving the same function in capsule development. And more to the point, the tomatoes don't have lignin, and so we're sort of curious what these genes are doing during tomato development. So just to orient you a little bit more to fruitful genes, this is basic angiosperm phylogeny, and the star shows the core eudicots there. And if you look in core eudicots, all core eudicots have two fruitful-like genes, which we put in these two clades called UFOL1 and UFOL2. But it turns out that in Solanaceae, there are additional duplications, and so there are actually a total of 4 genes. 2 UFOL1 genes, which we call FOL1 and FOL2, and 2 UFOL2 genes, which we call MBP10 and MBP20. So our project is aimed at looking at the function of these genes. This is just to show you the expression patterns of these genes in tomato on the top and Nicotiana, this desert tobacco on the bottom. I tried to actually brighten them up a little bit, but I can see it didn't work anyway. There are some very faint bands around in here and in here. Just to show you, these are the stages of fruit, carpel and fruit development here, carpel and fruit development here. And the genes, all 4 of these genes are expressed at some point during carpel and fruit development. So the structure of the project is that we're using CRISPR analyses, knocking out the single genes, UFOL2 and UFOL1 genes, and then all 4 of them, or at least this is what we are in the middle of trying to do at this point. We have all of the constructs made, we have all of the transformants, but we are now growing up the 2nd generation and hoping that we actually have the knockouts that we're looking for. And then we're also doing transcriptome sequencing, which is what I will be talking about now. We have the wild type data, and then eventually we will want to compare that. Oh, we'll want to compare the wild type data with the mutants to see if we can determine what genes are being regulated by the fruitful genes. And we're working with, as I mentioned, desert tobacco and tomato, but we've also decided to look at Solanum pimpinellifolium, which is thought to be the closest wild relative, on the theory that there may have been some changes that happened during the domestication process. So we're doing this looking at the transcriptomes over the course of developmental time, and so just to show you the different developmental stages, stage 1 in these fruits is more or less ovary development up until stage 2, which is fertilization, and fertilization kicks off this stage of rapid cell division. The patterns are different in the capsules and the berries. In the berries, you get both anticlinal and periclinal, and in the capsules, you only get anticlinal. And then stage 3, the cell division slows down, and then in the fleshy fruits, you get endoreduplication and cell expansion, and in the dry fruits, you get lignification. And then stage 4 is either ripening or drying down into hissins. And so the first thing that we did was we looked at, as I said, we were doing these wild type transcriptome studies, and so we attempted to look at these 4 stages in desert tobacco. We really have not been successfully able to get good RNA out of stage 4 yet. I think we're going to, basically what we're going to do is walk it back to the last possible day when we can get RNA. But we did get data for stages 1, 2, and 3, and you can see the comparisons that we were doing here. So between stage 1 and stage 2, and stage 1 and stage 3, we found, lots of nice differentially expressed genes. But when we compared stage 2 to stage 3, we found 9 upregulated, regulated, and 9 downregulated. And considering that you're going from the stage of cell division to lignification, this didn't really make a lot of sense to us. And we think what's going on is that you can see in this longitudinal section, you've got nice lignification going on here at the apex, but not yet at the base. And we knew this in advance, and so we did try to take just sort of from the apex when we were doing stage 3 and you know, at earlier from stage 2, but we strongly suspect that what happened was we didn't really get good separation of the stages. And so what we are going to do is, the student who's working on this, Alex, is going to do some in situ mRNA hybridization to try to identify when cell division is occurring and where cell division is occurring, and identify a really sort of refined area. And incidentally, I should say we've been doing this without the internal tissue, so no ovules, no seeds, no placenta, but just the pericarp. So in the meantime, we do have data for wild and cultivated tomatoes. So these are the comparisons, stage 1, stage 2, stage 3, and then 2 stages of ripening. The breaker stage where it first starts to turn color, and then the red ripe stage. And we're comparing them between species and then also between adjacent stages. And this artwork is by one of the students, Dinusha Maheeppala, who was working on this. And this is where sort of I feel like I have really kind of failed, in a word, because the students have been working on these analyses, and they got them, of course, to me kind of at the last minute, and it wasn't until I looked at them really carefully that I thought, this is not making a lot of sense. So this is in tomato here and wild tomato here, stage 2 and stage 3, so this is cell division, this is the cell expansion and endoreduplication. And basically, we really don't see a lot of difference. Most of the genes that are coming up are either cell cycle related or kind of basic metabolic processes. And we really don't see anything different. That may actually be true. That may be true in tomato because you have nuclear division going on here, and you have nuclear division going on here. You have various growth processes, etc. And so there may actually not be a lot of really distinct things going on. When we look at a comparison of stage 3, the endoreduplication and cell expansion, and the breaker stage, there we actually expect to see a lot of differences. But we're still seeing a lot of the same things coming up as being differentially expressed. We do kind of comfortingly see a number of things that are related to ripening in both species, although not necessarily exactly the same things. So I'm not like putting this in the bank at this point. I'd like to get some more, I want to talk to people who have a lot more experience with this type of analysis. And then here is one of the interspecific comparisons at stage 2, which is the beginning of cell division after fertilization, 123 differentially expressed genes, and again, I hate to say it, but kind of nothing exciting. Thank you. So one of the other students has been working on these co-expression analyses. He used this package in R, and he specified 15 patterns, and so this is what he found. And these are, that you see the full 1 gene, 2 gene is in this cluster, full 1 is here, MBP20 is here. MBP10 wasn't really differentially expressed, and so it doesn't come up in any of these. You'll notice that a lot of these patterns are actually pretty similar. And the one interesting pattern that I did find was that if you take these 5 clusters, this is half of the differentially expressed genes, all show a dramatic increase to stage 2 and then a dramatic decrease. So I don't yet know what these genes are, but it clearly indicates that something dramatic is happening at stage 2. Unfortunately, the patterns that he found for these fruitful genes don't really match the patterns that we got when we just extracted the data manually and looked at the expression over the course of time. So again, I'm not entirely sure what it is that has not worked out very effectively here. Almost done. He's also been doing some clustering analyses. Oh, this shows a lot better than I was afraid it would. And one of the things we noticed was that this is a late expressed gene involved in ripening, and the pimpinellifolium genes, the wild tomato genes, tend to have at least the same number, and in many cases more connections. So these are the fruitful genes. This is the CNR gene, and these are all the genes that are predicted to be connected to it. And the pipinellifolium genes seem to have more connections, except for the fruitful genes. So the full 1 genes, fruitful 1 and fruitful 2 actually have a lot more connections in tomato than they do in the wild tomato. So, okay, I'm now done. And so these are the conclusions that we have so far. We need to get better separation of the stages in desert tobacco, and that the genes are differentially expressed during tomato and wild tomato fruit development, but so far we haven't really found something that actually really tells us what's going on there. And stages 3 and 4 differ transcriptionally, but 1 and 2 and 2 and 3 not so much. And the 4th conclusion there you can see for yourself. So these are the people involved. Dinusha here and Alex are working on the transcriptome data. Ahsley was the REU student. This is, oh, here's Kevan here. Other lab members, this includes a lot of past members, so this is sort of the current lab members. Elizabeth has, what, 3 more days with us, I think, or something, and then she has a job. So, and if you have never seen the tobacco episode of The Simpsons, I encourage you, and then you will know why I put this here, because it's absolutely perfect. Thank you very much.